What are some of the basic sources of drugs? So you need to know that there are 6 sources of drugs. 1, plants. 2, animals. 3, minerals. 4, microbiological sources, and then also a synthetic or semi-synthetic, and then we can also have the last one, which is recombinant DNA technology. Very important that you know these sources. And so let's dissect, or let's expand on them. So let's talk about plant sources. So when you talk about plant sources, we classify them according to the part of the plant. So one thing you need to know is that when you're describing the sources of drugs, one thing you need to know is that depending on which part of the plant you get the drug from, or depending on what the crude drug is, you can have 2 categories. So you can have organized drugs and unorganized drugs. So organized drugs will be drugs that come from a particular part of the plant. So I mean things like leaves, flowers, fruits, seeds, roots, the bark, the stem. Those are organized. But then unorganized drugs refer to drugs that come from mostly the extracts, for lack of a better word. So what I mean is, for example, mucilages or gums. These are examples of unorganized drugs. So have we seen some trees? There are certain trees that release certain secretions on their barks. There's this tree that's actually called the bleeding tree. So it releases this red extract that almost resembles blood, except it's very viscous. So that's an example of an unorganized drug. So let's talk about plants. So when we talk about plants as being a source, we can classify, so I can give examples based on the parts where we extract the particular drug. So for example, when we talk about leaves, an example I could give you, I could start with eucalyptus, for example. Eucalyptus, you extract the oil, eucalyptus oil, and then it's used in cough preparations. And then we have Atropa belladonna. It's used to extract atropine, and then atropine is an anticholinol or an anticholinergic agent. So we'll discuss that in pharmacology. And then another plant where we care about the leaves, we have Digitalis purpurea or Digitalis lanata. So from Digitalis purpurea or lanata, you can get digitoxin, which is cardioactive, right? And then it's used in congestive heart failure. From the flowers, we can get Poppy papaver somnifera. What do we extract from there? We extract morphine. Another thing we can get from flowers is Vinca rosea. So Vinca rosea, the flowers, we can extract vincristine and vinblastin. So these ones are active in terms of cancer. So they can arrest microtubules. So they have anti-cancer properties. From the fruits, we can have cenapods. From cenapods, we get anthracene. Anthracene is a pegative. You need to remember it's used in situations of constipation. And then calabar beans. From calabar beans, we get a very important cholinergic agent that is called physostigmine or physiostigmine, however you want to pronounce it. And then from the seeds, we can have Nux vomica. From Nux vomica, we get strychnine. Strychnine is a CNS stimulant. Castor oil, we can get from the castor plant, or from the castor seeds, we can derive castor oil, and then that oil can be used as a laxative. From the roots, we can have, for example, from Rauwolfia serpentina, we can derive rezepine, and then the rezepine is a corticolamine depleter. So it's used in cases of hypertension. It has hypotensive action. So we'll talk about this one in pharmacology also. And then there's Apecacuana. So Apecacuana, what can you get? From the roots of Apecacuana, you can get emetine. And then emetine, just like the name suggests, it's used to stimulate emesis, right? Which means vomiting. And then from the bark we can have cinchona. So from the cinchona bark, we can derive 2 drugs, very common drugs. At least one of them is common. So we can derive quinine and quinidine. So quinine and quinidine are both antimalarial drugs. However, quinidine also has some antiarrhythmic properties, right? And then another thing, or another drug where we care about the back, we have atropa belladonna. So from atropa belladonna, we can get atropine. We discussed it. We said it's an anticholinergic agent. And then we also have the hyoscyamus niger, or hyoscyamus niger, however you want to pronounce it. From there, we can get hyoscine. Hyoscine is in the same class as atropine. We'll talk about those in pharmacology. And then from the stem, I only have one example for you. So we have Chondrodendron tomentosum. I know it's quite cumbersome to say, but it's a very important plant because you can get tobacurarine. So from tobacurarine, it's a very important drug because it's used in the treatment of tetanus, because it's a relaxant. It acts on nicotinic receptors and blocks them. So we'll discuss that in pharmacology. The other source is animals. So when we talk about animals, we can have the pancreas of animals can be used to obtain insulin, and then also human chorionic gonadotropin can be extracted from the urine of pregnant women. A cod liver can be used as a source of vitamin A and D. Stomach tissue can be used as a source of pepsin and trypsin. And then sheep thyroid, yeah, this one's a good one. It can be used as a source of thyroxine. And then the blood of animals can be used in the preparation of vaccines. And let's talk about the mineral source. So the mineral source, most of the halides have antiseptic properties. So iodine and chlorine will be used as an antiseptic. And then also in the treatment of iron deficiency anemia, you need to take note there are so many types of anemia. So in iron deficiency anemia, you can use iron. Then in the treatment of syphilis, you can use mercuric salts, even though we don't use these anymore because, or at least the use has declined, because usually mercury tends to be toxic, right? And then gourd salts can be used in the treatment of rheumatoid arthritis. Then we can also use zinc as a supplement, yeah, and many more. And then there's this idea of synthetic and semi-synthetic drugs. So when we talk about this as being a class, you need to know the difference between a synthetic drug and a semi-synthetic drug. So a synthetic drug is a drug where both the nucleus and the chemical structure has been altered, right? So you alter two things, the nucleus and the chemical structure. An example is emmetin bismuth iodide. And then when we talk about a semi-synthetic drug, the nucleus is intact, but all you've done is you've altered the chemical structure. An example would be Alexander Fleming was able to extract penicillin, right? And then penicillin had a downside. The downside is that bacteria evolved to produce an enzyme that could destroy the beta-lactam ring. So we'll discuss this in pharmacology, but just giving you an idea. And so what happened is that we had to make modifications to the structure of penicillin in order to make it resistant to those penicillinase enzymes, right? And so in order for us to do that, we had to add ARA groups to the penicillin core structure. So we haven't affected the nucleus, but we've just affected the outward chemical structure. And that's an example of a semi-synthetic drug. And so we created aminopenicillin. So we'll talk about this one and talk about antibiotics. I'll tell you how they were created and the modifications that were done, and it will make a lot of sense. So an example is ampicillin, right? Ampicillin is just penicillin. The only difference is that it has certain ARA group attachments that we'll talk about, like methicillin, nafcillin. All these are examples of modified penicillins. So we could give them as examples. If they ask you semi-synthetic drugs, we can have apomorphine, we can have methyltestosterone, ampicillin, nafcillin, methicillin. There are so many examples. And then we have the microbiological source. So under the microbiological source, it's showing how organisms can be a source of drugs. The classic example is the Penicillium notatum. So this was a mod that was seen by Alexander Fleming to produce the first antibacterial drug, which was penicillin, right? So from the Penicillium notatum, we can get penicillin. And then another thing you need to know, actinobacteria can produce an antibacterial drug that is called streptomycin. And then from micromonosporas, we can produce an antibacterial drug called gentamicin, right? And then lastly, we can talk about recombinant DNA technology. So recombinant DNA technology is it's a complicated process. Okay, I'll try to summarize the process though, because the idea is quite intuitive. So what we have is, we start off with a desired gene, a gene that we want. So that gene is located on a DNA strand, right? And so what we do is we need to cleave that gene, the desired gene that we want. And in order for us to cleave, we use an enzyme called a restriction enzyme, or sometimes it's called a restriction endonuclease. And so what we'll do is this: when we use that restriction endonuclease, it will cleave only that portion of the disease of the gene that we desire. And so that gene will be exposed, right? And then it will be taken up by a plasmid. And then we can get these plasmids and then introduce them into a culture of a particular organism we want to express this gene. So most of the times this organism will be a bacteria. So if we remember, bacteria exchange genes through three main processes. We have conjugation, we have transformation, and we have transduction, right? And so conjugation involves the use of a pilus. So you inject a pilus into another bacteria and then genes are transferred. Then transduction involves the use of bacterial macrophage. I think they're called bacteriophage, right? And then we have transformation. In transformation, this one happens when a bacteria dies, and when a bacteria dies, genetic material, usually due to lysis, genetic material can be exposed, and then another bacteria can integrate that genetic material into itself. And so this is what's going to happen. So this genetic material we've introduced to the culture, the bacteria will perceive this genetic material as being genetic material from, let's say, a dead bacteria or something, and then it will integrate the gene into itself. When it does that, this gene has the ability to code for a protein we want. Let's say we wanted to produce insulin, for example. So this bacteria will take in the gene for insulin, and then when it transcribes that gene, it will be changed into the protein insulin, and then you produce your drug. So that's a really summarized way of how recombinant DNA technology works. And so those are sources of drugs. Make sure you practice some of these. Make sure you get them, because you expect to find them in exams and tests. So that will mark the end of our video.